Hi, my name is Kalen Petersen, and I'm a postdoc in the ANSYS lab at Washington University in St. Louis. As persons with HIV live longer lives, new clinical questions about healthy and unhealthy aging have arisen in this population. Numerous studies have identified structural and functional brain changes associated with HIV positivity, although their degree and localization remain controversial in the CART era. These changes can resemble premature or even accelerated brain aging, which we define as the advent of older-appearing brain phenotypes at younger ages. Consequently, milder forms of neurocognitive impairment are thought to remain common, reducing quality of life, especially for older persons with HIV. However, the direct pathological and inflammatory effects of HIV are confounded by prevalent comorbidities. It is essential to quantify these risk factors. For example, cardiovascular burden can be assessed using the Framingham 10-year risk score for heart disease. Neurological findings in persons with HIV are also confounded by social determinants of health, since adverse socioeconomic outcomes are highly prevalent in populations with HIV. As in the ANSYS lab, we are learning to quantify social determinants of health alongside clinical risk factors. The map on the right reflects clear racial and socioeconomic divides in the city and county of St. Louis, Missouri. Warmer colors indicate Greater Area Deprivation Index, or ADI, a geospatial measure reflecting a composite of income, employment, housing quality, and other factors. Broad swaths of North and South City, as well as East St. Louis across the river, show sharp disparities compared to the affluent Central Corridor in West County. Here we test the hypothesis that social determinants of health, such as this, are related to brain aging. Additionally, we sought to measure the impact of adverse events in childhood and adolescence, such as exposure to violence or family instability. Therefore, we administered the Early Life Stress Questionnaire. Our data indicate that a greater proportion of persons with HIV experience 3 or more early life stressor events. To help us begin to disentangle all this complexity, we have utilized data-driven machine learning methods. In particular, brain-predicted age is a useful tool to quantify brain health from neuroimaging. It works like this. First, a machine learning model is trained on normative MRI scans to estimate participant ages from brain structure. The accuracy of this model can be assessed with cross-validation. Next, the model is tested on previously unseen data, including healthy controls and persons with a disease state of interest. Brain age gap is defined as the difference between estimated and true chronological age. If the model overestimates a person's true age, this positive brain age gap can serve as a marker of pathology. I look at the brain age gap as a conceptual lens that focuses complex neuroimaging data into a holistic, easily interpretable measure of brain aging. For this study, we utilized a well-validated, publicly available deep learning model called DeepBrainNet. Our group and others have found that the brain age gap is elevated in persons with HIV, at least at the level of group means, and that such differences are present in both gray and white matter. The graph on the left illustrates this effect, with true age on the x-axis, predicted age on the y-axis, and a widening gap apparent for people with HIV, shown in orange, compared to those without HIV in blue. However, you'll notice that within-group variability in brain-predicted age is high compared to between-group effects. This led us to ask whether such variability can be explained by factors besides HIV. We also asked whether the correlates of brain aging in persons with HIV are different from those in uninfected people. Finally, we sought to learn what spatial patterns in the brain were characteristic of elevated brain aging. To address these questions, we used a large sample of HIV study participants enrolled from the Washington University Infectious Diseases Clinic between 2009 and 2020. Participants underwent MRI and clinical characterization, and their residential addresses were used for geospatial measures. Serostatus groups differed significantly by age, sex, and race, so these factors were used as covariates in all subsequent analyses. First, we examined potential predictors of brain aging in isolation while controlling for multiple tests. Of the HIV-specific factors examined, detectable viral load and hepatitis C positivity were both linked with elevated brain age gap. Lower CD4 was also correlated with higher brain age gap, although this did not survive multiple comparisons adjustment. Of the factors available for both serostatus groups, the most clearly linked with brain aging in persons with HIV was cardiovascular risk, assessed using the Framingham score. We also observed a trend for a protective effect of educational attainment, although this was limited to persons without HIV and did not survive FDR correction. Having examined many potential predictors in isolation, we then sought to build multivariable models using best subsets variable selection. This method fits all possible variable combinations and selects the model that best explains the outcome variance while penalizing overly complex models with many predictor terms. The final model selected for persons with HIV contained 4 factors: Framingham cardiovascular score, hepatitis C positivity, area deprivation index, and early life stressors. Finally, we wanted to know what brain regions might be driving these effects. To better understand the neural underpinnings of brain aging, we sought to identify volumetric features salient to the machine learning model. As an approximation of feature importance, we correlated the brain age gap with regional volumes from FreeSurfer. We found that reductions in both gray matter structures, like hippocampus and amygdala, and white matter structures like corpus callosum were linked with greater brain aging, as were ventricular enlargement and white matter lesion volume. Interesting group differences also emerged, as persons with HIV showed greater associations between brain aging in specific cortical areas, like the precuneus, and subcortical areas, such as the basal ganglia, which may be informative for future studies. These data have helped us conceive of a model in which growing older with HIV, while experiencing comorbid disease burden and adverse socioeconomic circumstances, worsens chronic stress and possible inflammation. This, in turn, leads to increased aging-like processes in the brain, potentially producing cognitive impairment and diminished quality of life. I'd like to express my appreciation for my mentor, Bo Anses, the members of our lab, and my broader mentorship team at several great institutions. I would also like to thank our invaluable study participants and our funding sources at NIH. Thank you for your interest and attention.